welcome to SAM Biology class. Friends, today we shall discuss about expression vectors. A vector that is used for expression of a cloned DNA fragment in a host cell is called as expression vector. They are the basic tools in biotechnology for the production of proteins. E. coli is commonly used as the host for protein production, although other cell types may also be used for the same purpose. An example of the use of expression vector is the production of insulin that is used for the treatment of diabetes. An expression vector has features that any vector may have, such as an origin of replication which enable autonomous replication in the host, a selectable marker which is necessary for screening of the recombinant organism, and a suitable site such as multiple cloning site for insertion of DNA. The cloned gene may be transferred from a specialized cloning vector into the expression vector, although it is possible to clone the gene directly. Expression vectors used for protein production in organisms other than E. coli, in addition to an origin of replication suitable for propagation in E. coli, must have specific elements for maintenance of the vector in another organism. Such vectors are called as shuttle vectors. In order to express the cloned gene, the expression vector must have specific elements such as a promoter, a ribosomal binding site, a start codon, a termination codon, and transcription termination sequences. Friends, you know that the protein synthesis machinery of prokaryotes is different from that eukaryotes. Therefore, the expression vector should possess all the features that are appropriate for the host. Then only the gene can be expressed. For example, the prokaryote expression vector would contain Sime-Dahlgren sequence at the translation initiation site for ribosomal binding, whereas the eukaryote expression vector will contain Kozak consensus sequence for the same purpose. The promoter initiates the transcription and is therefore the point of control for expression of the cloned gene. The promoters used in expression vectors are normally inducible. That means the gene can be expressed only when required by introduction of an inducer such as IPTG. In some expression vector, the gene expression may also be constitutive. The expression vector is transformed or transfected into the host cell for protein synthesis. Some expression vectors may have specific sequence for transformation or insertion into the host chromosome. For example, VIR genes for plant transformation and the integrase site for insertion of DNA into the host chromosome. In addition, expression vectors may have specific targeting sequence that target the synthesized protein to a specific location, such as the periplasmic space of bacteria. After expression of the gene product, it may be necessary to purify the expressed protein. However, separating the protein of interest from the great majority of host protein can be a protracted process. To make this purification easier, specific purification tag may also be added to the cloned gene. The tag could be histidine tag, other marker peptides, or a fusion partner such as glutathione, S-transferase, or maltose binding proteins. Some of these fusion partners may also increase the solubility of some of these proteins. In addition, other fusion proteins such as green fluorescent protein may act as a reporter gene for identification of successful cloned genes. Expression systems for protein production by using expression vectors. Different organisms can be used as the expression system. In order to express the cloned gene, the expression vector should possess the elements which are appropriate for the host. First, bacterial system. E. coli is most commonly used as the host for protein production by using the expression vector. E. coli is useful for production of heterologous protein, which is relatively simple and convenient and also rapid and cheap. Other bacteria that are used for protein production include Bacillus subtilis. These vectors have promoters that are derived from lac operon or the T7 promoter. The system is regulated by lac operator. Examples of E. coli expression vectors are the PGEX series, where glutathione S-transferase is the fusion partner and the gene expression is under the control of tag promoter, and PT series of expression vectors that uses a T7 promoter. This figure shows the map of a member of the PT series of vectors, namely PT28A T7 lac OGAP. This expression vector has 3756 base pair. The important elements that are present in this vector are a T7 terminator, a kanamycin resistance gene and its promoter, an origin of replication, and the LAC-I gene and its promoter. In addition, this vector also contains a T7 promoter, a LAC operator, a ribosomal binding site, and the gene for green fluorescent protein. Yeast expression vectors. One of the yeast that is commonly used for the production of protein is Pichia pastoris. Example of yeast expression vector in Pichia are the PPIG series of vectors that have AOX1 promoter, which is inducible with methanol. Another yeast is Saccharomyces cerevisiae, which is used for gene expression studies. Another expression vector is baculovirus. It is a rod-shaped virus which infects insect cells. This virus is particularly used for the production of glycoprotein, although the glycosylation may differ from that of vertebrates. Next is plant expression vectors. Many plant expression vectors are based on the TI plasmid of Agrobacterium tumefaciens. The DNA to be inserted into the plant is cloned into a stretch of DNA called as tDNA that integrates into the plant genome. Last one is mammalian expression vectors. Mammalian expression vectors offer considerable advantages such as proper folding, post-translational modifications, and relevant enzymatic activity. Examples of mammalian expression vectors are adenoviral vectors, the PSB and PCMB series of plasmid vectors, vaccinia and retroviral vector, and baculovirus. Various mammalian cell lines such as Chinese hamster ovary, COS, including human cell lines such as HEK and HELA, are used for protein production by using the expression vector. Applications of expression vectors. Expression vectors in suitable host are used to produce proteins for research. Most of the proteins are produced in E. coli, but for glycoprotein and those with disulfide bond, baculovirus and mammalian systems are used. Expression vectors are also used for the production of peptide and protein pharmaceuticals such as hormones, vaccines, antibiotics, antibodies, and enzymes. In recent years, expression vectors have also been used to introduce genes into the plant and animal in order to develop transgenic organism. Finally, gene therapy is a promising treatment for a number of diseases where a normal gene carried by a vector is inserted into the genome in order to replace an abnormal gene or to supplement the expression of a particular gene. So friends, this is all about expression vectors. For more update, please subscribe my YouTube channel SAM Biology Class, and also please share your comments. Thank you very much.